3.2 Molecular Genetics. In the previous classes, we have studied classical genetics that focuses on principles of inheritance, or the way how genetic information pass on from parent to offspring or from one generation to the next generation. But now, in the second part of genetics, what we call molecular genetics, we will study chemical natures of hereditary molecules and how these chemicals determine phenotypes of organisms. Let's get started by defining chromosome. What is chromosome? Chromosome is a string of DNA that wrapped itself around histone proteins. Chromosomes are made from two chemicals, DNA and histones. What about chromatin? Chromatin is a complex of DNA and proteins that forms loosely coiled chromosomes within the nucleus of eukaryotic cells. When a cell is not dividing, the chromatin is loosely organized throughout the nucleus as loops of chromatin fibers. Individual chromosomes cannot be distinguished. The loose organization allows the genes to be active. As we can see from the diagram, DNA and histones are combined to form loosely coiled chromosomes within the nucleus, what we call chromatin. This chromatin is organized into certain number of chromosomes. So, chromosome is a string of DNA that is wrapped around histone proteins, as we saw from the figure. These chromosomes coiled more and form nucleosomes. Nucleosome means a basic unit of DNA packing in eukaryotes that consists of a segment of DNA wound in sequence around 8 histone protein cores. When this nucleosome supercoil, it forms solenoid chromatin fiber, which means a structure that shows 10 nanometer fiber of nucleosomes gets coiled upon itself to form 30 nanometer wide helix with 5 or 6 nucleosomes per helix. What is DNA? DNA is a double-stranded polynucleotide that is made from two antiparallel strands that wind into a double helix. Since it is polynucleotide, it is made from nucleotides. There are 4 DNA nucleotides. These 4 DNA nucleotides are deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytidine monophosphate. Each nucleotide is made from 3 molecules. These 3 main molecules are the 5-carbon sugar, deoxyribose, phosphate molecule, and nitrogen-containing bases. These nitrogen-containing bases can be purine bases and pyrimidine bases. Purine bases are simply a double ring base that include guanine and adenine. Pyrimidine bases are a single ring base that are thymine, cytosine, and uracil. But uracil cannot be used to make DNA nucleotides. Double antiparallel strands of DNA. How is a molecule of DNA put together? Do you remember from Unit 1 about DNA in different cells? That means in prokaryotic and eukaryotic cells. This organization of DNA with histones into a chromosome is found in all eukaryotic cells, but the molecules of DNA in prokaryotic cells are different in a number of ways. 1. DNA is much smaller and circular in prokaryotic cells, not linear as in eukaryotic cells. DNA is not associated with histones to form chromosomes in prokaryotic cells. DNA is a huge molecule made from two antiparallel polynucleotide strands. The nucleotides are held together by bonds between the sugar in one nucleotide and the phosphate group in the next. This bond is what we call phosphodiester bonds. The bond between deoxyribose and nitrogenous base is glycosidic bond. The nucleotides in one strand are paired with nucleotides in the other strand according to the base pairing rule by hydrogen bonds. Hydrogen bonds connect two strands of DNA together to create the double helix structure of DNA. So, the sequences of bases on the two strands are complementary with each other. That is, adenine always pairs with thymine and guanine always pairs with cytosine. DNA is a very stable molecule at normal temperature. This is because the sheer number of hydrogen bonds in the whole molecule ensures that the two strands stay in position. That means the individual hydrogen bonds that hold the two strands together are quite weak, but the sheer number of them in the whole molecule makes sure that the two strands can stay in position. The bonds that hold the nucleotides together, what we call phosphodiester bonds, in each strand are much stronger than hydrogen bonds and highly effective to make DNA molecule very stable. The stability of DNA molecule is very important to ensure that the genetic code does not become corrupted in any way.